えーこんにちは。本日は IBM テクノロジーが主催するセミナーにご参加いただき、誠にありがとうございます。まずは、本日進行を務めさせていただきます。株式会社ニューロサイエンスの足立と申します。よろしくお願いいたします。弊社は IBM テクノロジー製品の日本総代理店を行っております。本日のウェビナーでは、腫瘍微小環境ネットワークを標的とした新規治療戦略の開発と可視化をテーマに、東京科学大学の渡部徹郎先生と、KAISTのフィルハン・キム教授にご講演いただきます。ウェビナーの進行中にご不明な点やご質問がございましたら、Q&A 欄にご記入いただきますと幸いです。それでは、一つ目のセッションを担当する教授である渡部哲郎教授をご紹介いたします。今回のウェビナーでご講演いただけることに心より感謝申し上げます。渡部哲郎教授、それでは講演をよろしくお願いいたします。
はい、足立さん、ご紹介ありがとうございました。また、この、セミナーを、開催、していただいた IVM テクノロジー社に心より感謝いたします。また、IVM テクノロジーの、社長である、ピルハン・キム先生は、私の、長い間の、友人であり、様々な、日本と韓国の、研究の交流を通じて、非常によくしていただきました。改めて、ピルハン先生に感謝いたします。はい。それではですね、共有しておりますが、見えておりますでしょうか。よろしいですかね。はい。それでは、今回ご紹介いただきましたように、腫瘍微小環境をネットワークを標的とした、新規治療戦略の開発と可視化ということで、講演を始めさせていただきます。私はですね、東京科学大学の、医師学総合研究科病態生化学分野で、主に口腔がん並びに様々ながんの、研究をしております渡部と申します。よろしくお願いいたします。になります。当分野のメンバー、並びに、宮園先生、そして吉松先生、そして伊藤先生、そして、理研の白渦先生、そして、このシングルセル RNA シーケンシング解析に関しては G1 パーク先生にご協力いただきました。最後のスライドになりますが、我々はですね、今年の11月に、日本血管生物学会の学術集会を、東京科学大学の隣である、順天堂大学で開催いたします。ぜひですね、こちらにご参加いただき、血管生物、そしてがん研究について、この最新の、知見を紹介できればと思います。長くなりましたが、発表は以上となります。ご清聴ありがとうございました。
渡部哲郎先生、ありがとうございました。続いて、2つ目のセッションを担当する教授、キム・ピロハン教授をご紹介いたします。ピロハン教授はソウル大学で電気工学の学士、修士、博士号を取得され、その後、ハーバード医学大学とマサチューセッツ総合病院で研究員として活動されました。現在は韓国科学技術院、カイスト医科学大学院の教授として在籍されております。ナノ科学技術大学院の教授職も兼任されております。IBM テクノロジーの CEO 兼 CTO としても活躍されております。キム教授の研究分野は、バイオイメージング及び生体内可視化技術を含む様々な生命科学技術に及んでおり、多数の論文を発表されております。特に工学及びバイオテクノロジーの分野で世界的な成果を収められ、様々な学会で活発に活動されております。次のセッションに進む前に、1点ご案内があります。これからは英語での進行となります。恐れ入りますが、セッション中のご質問を英語でご記入いただきますようお願いいたします。日本語でご質問いただいた場合は、 セミナー終了後に、ご回答させていただきますので、よろしくお願いいたします。So, Firhan, please start your presentation.
Okay, so thank you very much. Thank you very much for everyone for joining today's webinar. So let me begin my presentation. So today I will introduce intravital microscopy for real-time in vivo visualization of cellular dynamics and angiogenesis and other immune cell interactions, mostly inside the tumor microenvironment. Okay, so I have a COI. And then so I think some of you already know about the intravital microscopy. So intravital microscopy is a technique that can provide this kind of cellular level, microscale images and video from the live and sized mouse model. So in this example, you can directly see the immune cell inside the blood vessel. It's neutrophil, actually, this red one. And then you can see how these immune cells are, moves around and then interact with each other in the live animal model, in the live tissue. Particularly in this example, it was obtained in the lung of an anesthesia mouse model. Well, in addition to the cell, we can also visualize the, presently labeled drugs. So as you can see in this video, this is the example, showing the delivery of the nanoparticle to the liver in vivo. So this is a real-time video, so we can directly see how the presently labeled nanoparticle is delivered through the blood vessel into the liver in real time. So to summarize, these two examples nicely shows that the intravital microscopy can acquire these live cellular level images and videos in a live in vivo animal model, also with some molecular and functional information. Okay, so for this purpose, the IVM technology, we produce this dual mode intravital confocal and two-photon microscope, as you can see in here. So it has a very compact size, and then everything is inside. So it has its all-in-one packagings. And then this system is optimized for in vivo cellular level intravital imaging of live animal model, as I showed in previous slides. And then this microscope can do both of two-photon imaging and confocal imaging using this single box. And then for each of the imaging mode, two-photon and confocal, we can obtain 4 process signal simultaneously. And then this microscope is using very high-speed laser scanner for real-time imaging with speed of up to 50 frames per second at 512 by 512 pixels per frames. So basically, we can obtain 50 images in 1 second with this pixel size. And then based on this real-time imaging capability, we can also provide automatic live tissue motion compensation functions. So let me just show you one example of, 4-color, two-photon imaging obtained from the live animal in live tissue. So this is one 4-color image data obtained from the skin of live anesthesia mouse model. So in here, in green color is actually the nuclei of the every cell. And then the red color is the cell membrane. And then blue color is the intravenously injected propofol, so it can nicely show the blood flow in blue color. And in white color, we can also see the collagen in the skin. Okay, so as you can see, we can nicely see the endocellular cell here, right? This is the endocellular cell nuclei, so we can see the capillaries, for example. And if you go deeper, we can even see the lymphatic vessel here. So this is the lymphatic vessel. And then also in the deeper area, we can see the larger vessels. And then this is another example. So this is another 4-color images obtained from the pancreas of the live anesthesia mouse model. So we can very nicely see this isthmus composed by the isthmus cells, right? As you can see in these images. And then we can also, obtain this level of imaging in the much wider area by taking the mosaic images. And then this is the automatically stitched wide area mosaic images obtained from the live animal in the pancreas. Okay, so this kind of live imaging can be obtained from almost every organ and tissue in a live animal model, as you can see in this slide. So so all these images were obtained from the live animal model, mostly mouse model. So all these bright dot is the single cells in the live animal model. So you can see many. So we can also have done a lot of imaging study use intravital microscopy to visualize the tumor. Okay, so to show some tumor imaging result, let me explain about our real-time imaging capability and automatic motion compensation. So the one challenge when you try to do live animal imaging is tissue motion because of the respiration and the cardiac pumping, right? So when you try to image the internal organ, we often see this kind of tissue movement in the live animal model, mostly due to the respirations. So basically, to solve this, we need two things. First, our imaging system should be fast enough to track all this tissue motion in real time. And after that, we can automatically identify the tissue motion and the level of focus. So we can select the well-focused frames and then do the frame-by-frame image registration to compensate this tissue motion. So this is the result. So if you can do that, even if from this kind of moving tissue, we can obtain this very well motion compensated cellular level images. So from this kind of movement can be automatically compensated and provided this high resolution, high SNR images. And then all this function is automatic. So this is our softwares. And then the real-time images obtained from the live tissue. All you need to do is just turn on the motion compensation function and press a single button, and then it automatically compensates the tissue motion and generate this motion compensated images in just a few seconds. Okay, then let me just show you some example of the tumor model imaging. So which can be combined with the Professor Watabe's investigation to improve our understanding about the cellular level dynamics and then the microenvironmental changes in a tumor model. So this is the live images. This is the in vivo image obtained from the CCOM of orthotopic syngeneic colorectal cancer model. So we inject this CT26 H2B GFP cells. So this is the colorectal cancer cell, and then we inject it into the CCOM. So this is the orthotopic model. And then we can, nicely, distinguish the individual, this cancer cell inside this tumor, as you can see in here. And at the same time, we can also, label the endocellular cell by injecting the CD31 antibody fluorophore conjugate, as you can see in red color. So we can nicely see the tumor vessel in vivo. And then also we can also see other, molecules, such as MECA79. So it's basically it's PNAD, one of the immune adhesion molecule. So it's very highly regulated in this tumor vessel, as you can see in here. And then for repeated longitudinal imaging, we can also use various imaging windows techniques. So let me explain one by one. So first one is the dorsal scaphoid chamber technique. So to save time, let me just show you this one. So we can permanently implant this metal window into the dorsal skin of the live mouse model. And then when we implant this window, we can inject the cancer cell in the middle into the subcutaneous tissue, as you can see in here. And then after the surgery, this mouse can survive up to 4 weeks. So we can repeatedly image these cancer cells in subcutaneous tissue. So let me show you one example. So in this work, we inject this MDA MB231 GFP cell. This is a human breast cancer cell into the middle of this dorsal scaphoid chamber. And then we let the tumor grow for 2 weeks. After that, we inject some nanoparticle that can target the activated tumor endothelium in this kind of tumors, including breast cancers. So this is the images we obtained at 2 weeks time point. So this green color is the breast cancer cell, okay? And the blue color is the tumor vessel. Again, we label this tumor vessel with the CD31 antibody. And then we intravenously inject the nanoparticle through the tail vein. And then we take multiple images at different time point, like a 2-hour, 6-hour, and 24 hours later after these nanoparticle injections. Okay, so let me remove the green color. So now we can nicely see the delivery of this nanoparticle first into the tumor vessel, like here, and then to the tumor cells later. So let me zoom in, so we can see the delivery of this nanoparticle through this blue color vessel, and then delivered to the tumor cell at 24 hours after the injections. So the beauty of using this imaging chamber is we can keep the mouse alive for a very long time, and then repeatedly observe the cellular dynamics at the same location inside this mouse model. We can do it with the orthotopic models, breast cancer model. So this is a memory tissue imaging window. So we can put the memory pipette, as you can see in here. And then also we can inject any type of breast cancer cell to create the syngeneic model. So this green color is actually 41 breast cancer cell. And then as you can see in here, we can nicely see this breast cancer cell and the vessels, the tumor vessels over time. And then let me move on to the other technique, abdominal imaging window. Basically, this ring-shaped window, again, it can be permanently implanted on the abdomen or side of the mouse model, as you can see here. And then after that, after this implantation, again, we can keep the mouse alive for 3 to 4 weeks, and then repeat the images. So this is our result. So we implanted this window on the side of the mouse model to see the kidney, as you can see in here. And then under our microscope, we can visualize it like this. And then we can find the single kidney tubule, the renal tubule, as you can see in here, from day 0 to day 7. And then we can repeatedly find the same location and then see what happens in pathologic condition or normal homeostatic conditions. Let me move on to another example. So in this case, we implanted this imaging window, abdominal imaging window, to see the pancreas, as you can see here. So this whitish tissue is the pancreas, and this red one is the spleen. And then we can inject the process cancer cell, panco2GFP cancer cell, into the pancreas, as you can see here. And then we can keep the mouse to be alive for more than 2 weeks to 3 weeks. Like this, and then we can see this growth of the pancreatic cancer cell. And then let me zoom in. So if we can zoom in, then we can see this green color, pancreatic cell growth and the infiltration into the other areas, as you can see here, right? And then if we zoom in this infiltrating area, we can see this cancer cell, infiltration inside the pancreatic cancer pancreas. And then also we can very nicely track how the tumor vessel changes over time, as you can see here. So another example. And also other phenomena, other cancer progression can be also imaged. This is some ongoing work. So in here we use this PDX1 Korea related to KRAS G12T mutant mouse model. So in this mouse model, after tamoxifen injections, some of the pancreas cell, such as alcinite cell, can have KRAS mutation, KRAS G12D mutation. At the same time, it starts to express RFP. And then after that, we observe every 2 days, and then we find out this early, alcinite to ductal metaplasias. So if you can see here, these alcinoids changes every 2 days like this, and then form these large ductal-like structures. So by using intravital longitudinal imaging, we can nicely track these changes in a live animal model. Okay, and then finally, this cranial imaging window I'd like to explain about this cranial imaging window technique. So basically, we make a hole in their skull. And then we put the transparent cover glass. And then we protect the all the exposed skull with the dental cement. After this surgery, so we can nicely see the brain for more than several months to even a year. So it looks like this. So we can manipulate the mouse model again, and then put it into our microscope. And then through this cranial imaging window, we can nicely see the dural, subarachnoid space in the meninges and the brain cortex like this. And then by using some transgenic mouse model, the NG2 mouse model, we can nicely see the dural, and then we can also see the arterial, capillary phase site and the venule in the live mouse brain. And then also we can use this cranial imaging window technique to evaluate the delivery of the drugs across the blood-brain barrier. So this is the one example. So the upper panel is non-modified antibody drugs, and the lower panel is modified antibody drugs that can more efficiently penetrate the blood-brain barrier. Okay. So this is the, again, longitudinal imaging after the drug injections. So you can see, compared to the control, this modified antibody drug can very nicely penetrate the vessel, right? And then infiltrate into the brain, as you can see here. So the intravital microscopy is, again, it's very useful for drug PK and PD analysis and validations. And then also, other, glioblastoma model. This red color is glioblastoma cells. And then again, we take imaging, every 12 to 24 hours, and then we can find out how this, GBM cells grow over time, and how this, the vessel, changes in just 3.5 days. Yes. So I only showed a few, but for various organs, we provide these imaging window solutions for longitudinal, in vivo imaging of a single mouse model. And then as you can see, as you saw in the previous slide, it can be very useful to study the very dynamic, tumor microenvironment in a live animal model. Okay. All right, so I only showed this one, but we recently made more open space versions like this one. So with this one, the FS model, we can image the larger animals such as rat or parrot. And then even in the lab, we can, obtain these, cellular level images in all different organs, such as heart, liver, and kidney in the lab. And then even for the larger animal, we recently make this robotic arm system, robot systems. So our microscope is miniaturized, attached into this robot, and you can put it anywhere like this, and then obtain this, cellular level images in the large animal, such as non-human primate or pig and dog. And then also imaging software, image analysis software is also very important. And then we provide this IVM studio software, so it can handle this very large area images, this whole brain slides, automatically stitched. And then we also provide this adapter for common normal slide images, as you can see here. And then we can also deal with other live samples, such as organoid sample, using on-stage incubator. And then this studio software can deal with large size 3D imaging data, as you can see in here. So we can perform the 3D rendering and then do the analysis. And this is a 4D data. Basically, we obtain the 3D images at different time points, and then see how this individual cell moves around in 3D space. Okay, to summarize, intravital microscopy is a technique for microscopic scale imaging and analysis in a live animal model in vivo. And then in various organs, we can obtain these dynamic cellular level images and videos to observe, to analyze the cell trafficking, cell to cell or cell to microenvironment interactions, all in the live animal model. And then we can also use intravital microscopy to study the drugs, right? To test the drugs. So proprietary labeled drugs such as nanoparticle, antibody drugs, or peptide drugs can be very nicely tracked by using intravital microscopy. So this is the end of my talk. So if you're interested in this intravital microscopy technique, so please visit our webpage, www.ivimtech.com, and then you will have all the information about our technology. Thank you. Thank you very much for your attention. Okay, then now we can move on to the Q&A, I believe. So in the Q&A chatting, I can see one question. The question is, based on your experience, what aspects of immune cell vascular tumor cell interaction observed by intravital imaging differed most markedly from those seen with ex vivo or static imaging method? Okay, that's very good questions. So in here, if you see very carefully, you can see this one capillary in here, and then we can see this green GFP positive breast cancer cell plunged into the this vessel, right? So this kind of tumor cancer metastasis event, the cancer cell need to invade intravasate into the capillaries or larger vessel, and then plunge and then extravasate. So those dynamic process of the cancer cell movement into the vessel and then from the vessel, we can use intravital microscopy to capture the movement and then to observe the some molecular picture involved in those intravasation and extravasation in the context of the metastasis. And then one of our observation also find out some immune cell can modulate this endocellular cell to further improve those kind of intravagations moment or extravasation moment. So basically by using intravital microscopy, we can see different cell types simultaneously, dynamically. And then so we can see endocellular cell, we can see tumor cell, we can also see various immune cells such as macrophage, and then dendritic cells or other immune cells, right? So we can basically we can see all these different cells simultaneously and then see how they interact with each other. And then one of the interesting thing I saw was their dynamic interactions during the process of extravasation and invasion, intravagations. Okay, I hope I can answer your question.
So Pilhan?
Oh, yeah, thank you. Yes.
Yeah. Can I ask a question?
Sure.
Yeah. Thank you. Thank you very much for wonderful presentation. As always. So I think it will be really nice. If we can observe. Like you just showed, observe the cancer cells departing from the primary tumor and metastasizing to secondary organs such as liver and
Yes. Yeah.
real time. So my question is, is it possible with your excellent microscope system to simultaneously image two organs in single amounts? Or
Okay.
are you planning to develop such a system in the future?
Okay. For now, normally we only image one organ at one time. Because in many cases we use this imaging window that can specifically implanted on the one organ, such as pancreas, kidney, or liver. So it's a little bit difficult to observe the two organs simultaneously in a single mouse model, but still we can do sequential imaging. One organ, and then later we can see other organ in a single mouse model. And then if you look at the size of the mouse model and then our imaging setup, it may be a little difficult to see the two organs simultaneously. But we can do, as I told you, we can do it sequentially. So one organ and the other organs.
Yeah. That's
So one example we have done is we can, inject the cell into the spleen or other, other periphery, the primary tumors, and then see the, metastatic organs such as liver, as you just explained.
yeah. Since when tumor progresses, the such tumors may influence the structure of our blood and the lymphatic vessels in the
Yes.
so maybe your suggestion of sequential observation in the 6 months may greatly help or resolve such changes. Thank you very much. Yeah.
Yeah. Thank you. Okay. Oh yeah, there's one more questions. So I hear the ethical regulation for mouse experiment is getting severe worldwide. Yes. Could you please comment on how you can perform intravital imaging in Korea? KAIST, for example, how long can you image one mouse continuously under anesthetization and CGI in your license? Well, so we had their approval from IACUC for long-term imaging under anesthesia up to 3 to 4 days. So, but if we want to, do more longer imaging, then I think we can apply for the, approval. The important thing is to minimize the, pain felt by the mouse model. So, it's generally accepted. If the mouse is anesthetized during the imaging, then it's okay to increase the imaging period as long as the mouse is anesthetized, and then pain relief drugs is continuously supplied. And even after the imaging, we still need to do our best effort to reduce the pain, such as by giving the proper pain relief drugs, and then also provide the nice conditions and nice environment. And then of course, all those intravital imaging experiment needs to be approved in advance by the ethics committee. But specifically for your questions, as long as the mouse is anesthetized, we normally don't consider it as painful procedures. So the short answer is we have experience of 3 to 4 days, 4 days continuous imagings on the anesthesia, and then I'm kind of sure that we can get another approval if we want to see even more. And then another practical thing we need to consider is for 3-day or 4-day imagings, we also need to provide nutrient and water, continuously to the mouse model. And then the easy way to do that is using the syringe pump, and then put the test the tube inside the peritoneum. So slowly inject the nutrient, such as glucose and other things, to the peritoneum using a syringe and syringe pump.
Thank you, so Pilhan.
Okay.
Okay.
So if you have any more, specific question later, please send us an email at any time. So in my slide, here. So please send us an email, any questions to this information@ivimtech.com, or the Adachi sensei, Adachi-san
Okay.
in Japan.
Okay. Thank you. これをもちまして、ウェビナーを終了いたします。本日はご参加いただきありがとうございます。改めて感謝申し上げます。ウェビナー終了後に簡単なアンケートが表示されますので、追加のご質問等ある方はご回答いただければ大変助かります。回答は後ほどメールにていたします。どうもありがとうございました。
りがとう
ありがとうご
ございました。
ざいました。
Thank you very much.